In this lecture on our module on special topics in protein chemistry, we are going to be looking at enzyme action and proteolytic cleavage. From this, we will be looking at the studies of proteases, Edman degradation, what we mean by enzymatic cleavage, and specific examples of sequence determination. In the previous modules of 6 and 7, we have looked at enzymes, enzyme classes, enzyme mechanisms, kinetics and inhibition in detail. However, what we will look at here is how to fragment a specific polypeptide chain and by that determine the specific peptide sequence, the polypeptide sequence associated. So we will be looking at enzymatic cleavage and also some degradation methods into what we would want to do in automated peptide sequencing. So the amino acid composition and the sequence determination methods are important. The amino acid composition tells us how many of each amino acid types are present, and the sequence determination methods tell us exactly in what sequence those amino acids are. So if we want to determine the total amino acid content, this can be done by complete hydrolysis of the protein and identification of the specific amino acid residues present. If we want to look at now methods of chemical and enzymatic cleavage, we can do this hydrolysis of the peptide sequence with greater than 6 molar or greater than 2 molar TFA, trifluoroacetic acid, and we have the following effects associated with this, which we also have to be aware of. This could destruct our tryptophan residue altogether. This could result in the partial destruction of the serine and the threonine. And because of the harsh methodology used, we could have a conversion of the amide amino acid residues, that is asparagine and glutamine, to their respective acids. So based on this information, we would then have to construct our corresponding peptide. So if we look at chemical and enzymatic cleavage, we can look at Edman degradation in automated peptide sequences. In this case, the N-terminal of the peptide can be analyzed by Edman degradation using what is called a PITC and PTH derivatives. And we have Sanger's reagent also, that is 1,fluoro,2,4-dinitrobenzene or dansyl or dabsyl derivatives that also can be used for N-terminal detection. Without going into much detail of this, we will just have a brief overview of how these work. So the Edman degradation procedure, it labels and removes only the N-terminal residue from a peptide and leaves the other peptide bonds. So it can identify the N-terminal residue of the polypeptide and the others can be purified and recycled for the Edman process, and from that we can then determine the sequence of the peptide, because each time we will now have a new N-terminal because the previous N-terminal has been removed by the Edman degradation procedure. In this experiment, the peptide bond nearest to the amino terminus of the protein is cleaved in 2 steps. They are carried out under very different reaction conditions. One is basic and one is acidic, and each step has to proceed to completion before the second step can be initiated. So this is the reagent that is used, PITC, and here is our peptide and here is our N-terminus, and the rest of the peptide is along this direction. We know that N-terminus is the N-terminal residue is the first residue in the protein. The reagent is phenyl isothiocyanate. We have a specific reaction where in the reaction under the basic conditions, we have the formation of an adduct, that is the phenyl thiocarbamyl derivative. From that, we have a specific reaction that is going to give us, under conditions of heat, a peptide that is attached to our PITC. So we have the PI derivative here, and we have hydrolysis, and we have the connection in a manner that is going to be resulted of resultant peptide that is going to be cleaved from the specific residue attached to our phenyl thiohydrantoin derivative. So what happens here is initially we have our overall peptide given by this, and we are now concerned with the specific amino acid residue, that is the first amino acid residue in this sequence. In the process, what happens is under these basic conditions, the phenyl thiocarbamyl derivative is formed, and once this is formed, there is an adduct with the peptide connected to it. However, this is then hydrolyzed where the peptide containing part with the new amino terminal, this is the new amino terminal, is cleaved off, and we have the derivative of the amino acid residue that was the first one in the sequence. This cycle can go on and we can get the sequence, the N terminal, or we can get amino acid number 1, then 2, then 3, and so on and so forth. In the Sanger's method, there is a sequence of amino acid residues in the polypeptide chain. This was first worked out with insulin. And here the amino terminal is also labeled and its identity determined. This can be labeled with DNFB, dancyl chloride, or dapsyl chloride, which is going to give us the first amino acid in our sequence. So when we are looking at peptide end group analysis, in this case, the sequence is determined by labeling the N terminus of the residue. So here is our peptide where we have our specific amino acids given by their side chains R1, R2, R3. The reagent in this case is our DNFB, dinitrofluorobenzene, and we have a specific reaction again go on here where we have this as connected to our polypeptide chain now. This will then be hydrolyzed. After the hydrolysis, we will have a connection with the first amino acid residue with our derivative that can be monitored. And from this, we can determine what is R1. So, what other methods are available? We have the proteases. Proteases is a topic that we covered when we looked at enzyme mechanisms in terms of the workings of chymotrypsin. And we know that this also cleaves peptide bonds by hydrolysis in an enzymatic fashion. And we have a classification based on the catalytic residue, like we looked at serine proteases, aspartic proteases, or cysteine proteases, depending upon what is present in the active site. So the specific enzymatic cleavage that we now look at is the protease acting on the peptide bond, resulting in the cleavage of the peptide bond. So this means that if we have a serine protease and we have our specific protein sequence, our polypeptide sequence, the chymotrypsin is going to fragment it in a fashion that will selectively cleave at the carboxyl end of aromatic side chains. We have trypsin that will selectively cleave at the carboxyl site of arginine and lysine, provided it is not followed by a proline. Similarly, we can have elastase that will cleave at the carboxyl site of valine and threonine. So let us look at how this action occurs. We have looked at the mechanistic details. We are just going to now look at how we can determine the overall polypeptide chain or the identity of a peptide in the specific order in which the amino acids are by the usage of enzymatic cleavage and chemical cleavage to give our peptide sequence. So we know that this selectively cleaves at the carboxyl side of aromatic side chain. So given that this is our peptide sequence, we now realize when we look at this that we have two aromatic side chains here. But this cleaves at the carboxyl site. So this arrows indicate the N site, because we know that our polypeptide sequence is in this fashion. So this is the N terminus and this is the carboxyl terminus. So at the N terminus, this cleavage site is not correct. However, if we move them over again, we do not have the correct amino acids in this case. So still there will be no cleavage. However, if we move it to the carboxyl side of the aromatic side chains, in this case we have two of them, the tryptophan and the phenylalanine, we will result in this cleavage. So then we can now follow an Edman degradation or a Sanger method for the peptide fragments and determine the N terminus of each of these fragments into trying to figure out what the original chain was. So this is the methodology where we can use chymotrypsin. Similarly, we can use trypsin. In the trypsin method, we find out that this selectively cleaves at the carboxyl site of arginine and lysine. So in this case, if we try and identify where we have our lysine and arginine, then we would know that this will cleave at the carboxyl side. So this is not the carboxyl side, but it is the N-terminal side of the specific amino acid. So we would have to look at the specific cleavage sites, giving us our specific fragments in this case. Again, we can look at the identity of the N-terminus, or we can, now that we have a phenylalanine, we can now look at this specific fragment and look at another enzymatic cleavage mechanism. Similarly, with elastase, it cleaves on the carboxyl side of valine and threonine. So we would have to know if we have a valine threonine present, then we can actually look at the specific C-terminus cleavage and find out that we have our cleavage. So now if we look at the same peptide and use these different enzymatic methods, we will then be able to piece together our results to find out the overall peptide sequence, which we will see. In the case of pepsin, this also selectively cleaves at the amino site of leucine, phenylalanine, tryptophan and tyrosine, provided it is not after a proline. So if this is after a proline, so there will be no cleavage here. However, we will have these fragments formed. So this is the methodology by which we can actually determine the ideas associated with the experiments. So we have another mechanism. This is a chemical cleavage that will tell us this works on methionine in the formation of what is called a lactone. Now we have our peptide. So this is the N-terminal side of our protein polypeptide chain, and this is the C-terminal side of the polypeptide chain. And we know that we have our specific peptide bond, and this is the methionine side chain. In this case, we have the CNBR, the chemical cleavage that is going to occur in this case, where we have a specific reaction occur in the elimination of the bromide ion, and we have an adduct formation. Now, in this case, when we have now hydrolysis, we have a cleavage form here. This is where we have the derivative from the methionine. The sulfur comes from the methionine in this case, and with hydrolysis we actually have a lactone formation with this fragment of the peptide, and the rest of the fragment of the peptide, the rest of the chain, the polypeptide chain with the new amino terminus, has been cleaved. So what this does, CNBR will work on a methionine residue. So the point where we have our methionine residue followed by the other polypeptide. So this is the N terminus and this is the C terminus of this. So this is the C terminus and this is the N terminus of our polypeptide chain. If there happens to be a methionine, we will have a lactone formation at this position. And depending upon how many methionines we have, we will have lactone formations at each of those positions, resulting in a specific number of fragments, depending upon how many methionines are present. Similarly to the case of the enzyme case, where we'll be looking at the cleavage fragments from a triptych digester, as it is called. Carboxypeptidase works on another hand, where it cleaves at amino site of the C-terminal amino acid. So we have an identification for the N-terminus using the Edmonds or the Edmond degradation method or the Sanger method. Now we want to know what is at the C-terminal amino acid. So this case we can use an enzymatic cleavage. For this there are several types. We have the carboxypeptidase A, B, C, A, B or D rather, that have specificities, because we have to remember that the enzymatic cleavage means we have to consider the substrate specificity, which is also important. So when we have a selective cleavage at the amino site of the C-terminal amino acid residue, it means it is just before the end. So this is the amino site, and we can identify our C-terminal amino acid. Similarly, if we used an Edmond degradation method, we could find out the N-terminal amino acid for this specific polypeptide sequence. Then we can use other methodologies. For example, if we use the trypsin, we would have a link break here. And then based on this, we would be able to identify or work out what our peptide sequence is. So we have the amino acid sequence determination methods that have the Sanger method, the Edmond method, and the C-terminus method that can be used with the carboxypeptidase. So we have one end, that's the N-terminus, that can be determined by these two methods. We have the carboxypeptidase method that can be used to determine the C-terminus, and the CNBR that we looked at that will cleave up the peptide bond after the methionine residue. And there are specific other chemical cleavage methods that can work on our polypeptide chain. So if we have now an example where we are looking at a tryptic digest of a heptapeptide. What we mean by a heptapeptide is that there are 7 amino acids, and it has been found from, say, a complete hydrolysis of the polypeptide chain that there are 3 lysine residues, 2 methionine residues, 1 serine and 1 proline, and there obviously there can be several sequences in which they can occur. But the tryptic digest of the heptapeptide, consisting of this combination of amino acid residues, gave the result was a tripeptide and a tetrapeptide, which meant that if we have a sequence where we have 1, 2, 3, 4, 5, 6, 7, we could have a cleavage here that is going to give us our tripeptide and tetrapeptide in one case. So this would be case 1, or case 2, where we would have the cleavage here giving us a tetrapeptide here and a tripeptide here. So the identification is to where this specific amino acid is going to cleave and what we would expect. So we know that the trypsin will work after the lysine residues. So lysine is K. So we could have a break here for this particular sequence in the identification of this peptide. If you look at number 1, we can have a cleavage here, we can have a cleavage here, but none of this is giving us a tripeptide and a tetrapeptide. We could, in this number 2, we could have a cleavage here and a cleavage here, but again we will have a single amino acid and a dipeptide, so we do not get a tripeptide or a tetrapeptide. In this case we have a K here, but we see that it is followed by a proline, so this means that the cleavage will not occur here. We will have a cleavage here, which gives us a tripeptide and a tetrapeptide. In this case also, a cleavage here and a cleavage here does not give us the tripeptide and the tetrapeptide. So this is where we have our answer, in which of the sequences shown is going to conform to our experiments here. So now if we have another example where we are looking at a multimeric protein with 4 identical subunits, and we want to do an Edmond degradation on it. But since there are 4, so what should we do? Now, is it necessary for the 4 subunits to be sequenced individually? Should the cleavages done be to look at the 4 sets of fragments? Do we need to look at the peptide bonds broken, or do we need to actually disrupt the disulfide bonds? Now, since the 4 subunits are identical, it means that the 4 sequences are also identical. So each of these are going to have 4 they're all the same. So if we just take one and do the peptide fragmentation, the enzymatic cleavage, we will be able to identify the sequences for all of them. But we see that we have a connection here between these two subunits. So given our methodology in the enzymatic cleavage, this disulfide linkage would remain. So we would have a different peptide fragment for this particular set of subunits. So in this case, what would be necessary would be to break the disulfide bonds with beta mercaptoethanol, as we saw, so as to get exactly the subunits that can be sequenced in several methodologies. So whether we are using a chemical cleavage method with these different reagents, which are going to be used for specific ways in which we can identify the amino acid residues associated here, or if we use enzymatic cleavage methods, where we saw we can use trypsin. There are other methods. Here's clostridium, staphylococcus protease, a V8 protease that cleaves on the carboxyl side of aspartate and glutamic acid and can be used to cleave only for glutamate under specific conditions where we have ammonium ions. Then we can also look at enzymatic cleavage for thrombin, for chymotrypsin, for carboxypeptidase as we saw, and also for elastase and pepsin. There are many, many enzymes we understand that can cleave these peptide bonds at specific locations. Given that there are enzymes, we understand their specificity associated with their mode of action, and based on this we can look at the identification of peptide sequences. So here we have the V8 protease, which, as was mentioned, it can cleave on the carboxyl side of the aspartic or glutamic acid, and in the presence of ammonium ions it is limited to glutamic acid. Again, we have another method, a diapsyl chloride. This is again a labeling agent, chromophonic labeling agent that is used in HPLC for derivatization of amino acid residues, and it reacts freely with all amino acid residues and gives us photostable derivatives. So this can also be used to identify the N-terminus. So let us look at how these experiments can actually be done. So if we have a deadly, say a spider venom that has an unknown polypeptide sequence that needs to be analyzed, we have to do some specific chemical reactions, specific enzymatic reactions, and this is what is determined. We have the amino acid composition. This is determined by a complete hydrolysis where all the peptide bonds are broken and we can identify that this is what we get. Okay, it has an unknown polypeptide chain which we are going to determine the sequence. The treatment with CNBR showed no effect. The treatment with chymotrypsin gave 2 tetrapeptides. Treatment with diapsyl chloride yields diapsyl histidine. Then treatment with trypsin gave 1 tripeptide, 1 tetrapeptide, and free methionine. So based on all this information, we are now going to work on each of the points to find out what the overall sequence is. So this gives us an idea of how to identify from the polypeptide sequence, from the specific enzymatic or chemical reactions that are done on the polypeptide to determine what the sequence are. So we have 8 amino acid residues. Now, from these 8 amino acid residues, we want to identify what we have. Now, so this we have the 8 residues that was determined from the first point. The treatment with CNBR showed no effect. We understood from the previous slides or the previous part of the lecture that CNBR acts on methionine to form the lactone. Now, the lactone possibility is only when the methionine is within the protein or within the polypeptide chain. This means that when we have the treatment with CNBR showing no effect, it means that methionine has to be the C-terminus, right? We can also determine the C-terminus by using a carboxypeptidase. The next was treatment with chymotrypsin gave us 2 tetrapeptides. Now, chymotrypsin we know so we have the amino acid composition that will tell us now where the chymotrypsin will work. And we get the chymotrypsin, we know, that works on the carboxyl sides of aromatic amino acid residues. So it gave two tetrapeptides, and given that this is an octapeptide, we know that we can have the cleavage here. So that means that we have now our two tetrapeptides. The dapsyl chloride gave us dapsyl his. So what does this mean? This means that this is giving us the first amino acid. So we have histidine here, we have the tryptophan here, we have the methionine here. Now we have to identify the others. So we look at treatment with trypsin. Trypsin will work, we know, after amino acid residues that are basic. So we have arginine and we have arginine here. So the arginine gave one tripeptide, one tetrapeptide. So the free methionine was here. So this had to be preceded by an arginine. And we had one tripeptide and one tetrapeptide. So given that, we could look at the location of the arginine based on the identification of the specific residues. So given that the location of the arginine elsewhere would not give us the tripeptide and the tetrapeptide and the free met, this is the only location possible for the arginine residues. It's like a puzzle. So we can identify then, so the treatment of V8 protease gave two dipeptide and in the presence of ammonium, so which means that it would work only on glutamic acid. In this case, we do not have an aspartic acid, so it does not matter. So in this case, we have two dipeptides and one tetrapeptide. So based on that, we will work out the locations again and find out that the cleavage would give us with the V8 protease, give us the rest of the sequence. We have only one amino acid left, that is the serine. So we have the sequence of the venom peptide from all these specific chemical and enzymatic methods that are going to help us identify the sequence. Similarly, so this is the overall summary of what we looked at and how we identified the specific by looking at chemical methods of cleavage, by looking at enzymatic methods of cleavage to determine the overall peptide sequence. So let us look at just another example, a similar example where we have the hydrolysis of the peptide sequence and amino acid analysis that gives us this set of now amino acid residues. Now the fact that we have this, it may so happen that we have two of one of them. Two, it does not necessarily mean that we have one each. That is my point. So the treatment of the peptide sequence give PITC yields M. Treatment with carboxy peptidase gave Q. And treatment with protease gave two products. And the E containing product, tripeptide, had sulfur. And treatment with chymotrypsin gave two products, one of which is a dipeptide. So again, we want to find out what the sequence of the peptide is. So if we work in a similar analytic fashion like we worked in the previous case, so we have KMCEF, which means that this is a pentapeptide but could have more than 5 residues, but these are the specific identities of the amino acids presence. We have the Edmond degradation gives M, the N-terminal residue. The carboxypeptidase gave Q, that means the C terminus in Q. And after hydrolysis of the peptide, E was obtained. Thus, Q is hydrolyzed to E. So, this means that there is another additional amino acid E. So, the Staphylococcus protease cleaves after D and E. Therefore, D or E has to be present. E has been obtained by hydrolysis of Q, which indicates that this is not a pentapeptide but an hexapeptide with an additional E, because after hydrolysis of this, we also got E. So working out on this mechanism, we have the E containing tripeptide that contain the sulfur. So the other residue is also a tripeptide. So we worked out the two different tripeptides, MXE and X1X2Q. This is our hexapeptide. And this was the one that contained the sulfur. So if we work it out with the different methodologies and the different analytical methods, it would be a good idea to look at the original problem and then try to work it out as to how each of our methodologies in terms of the enzymatic cleavage and the chemical cleavage fit into an understanding of the overall sequence and how it is to be identified. So this can work out to give us the original sequence where we have the tripeptide MXC and X1, X2, XQ. These are the ones that we are trying to identify the locations of the specific amino acid residues. And since one of the tripeptides that had the methionine or had the E had the sulfur, indicated that X had to be cysteine. So this gives us our identification. So we get the N terminus, we get the C terminus, and we get the specific fragments with the enzymatic cleavage. So from the enzymatic action and the proteolytic cleavage using proteases, Edmond degradation, enzymatic cleavage, we can actually look for a sequence determination. There are methodologies now that have automated peptide sequencers where you can actually give your peptide or your protein. There would be a tryptic digest. From the mass of the tryptic digest and following the sequences and following specific chemical cleavage and enzymatic cleavage methods, we can actually determine the specific sequences of the peptides pieced together by different methodologies and then find out our overall polypeptide sequence. These are the references. Thanks.